This lesson we're going to take a look at the similarities and differences between diffusion and osmosis. First thing to keep in mind when we're talking about either diffusion or osmosis is we have to remember that molecules are always moving. They move randomly and then bump into each other and to barriers. So if we have any molecules, they're not going to follow a predetermined or a set path. They're going to move randomly and they're going to continue to do so. So keep that in mind as we go through the following definitions. Defining diffusion. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. So if we look at this diagram here, we have lots of blue molecules on the left-hand side. They're tightly packed together, so we consider that molecules are in high concentration. They're going to move towards the left, the area where there are not a lot of molecules. They're in low concentration. There's lots of space between them. Another way to show that is by a time segment. So T1 being time 1, time 2, and time 3. We have a high concentration at the center here. Those molecules are going to want to move away from each other and get to an area of low concentration, which is this area farther out from the center. So if we look at this time 1, they begin to move away from each other. Time 2, they're continuing to move away from each other. And time 3, even still moving away from that high area of concentration in the middle to the low area of concentration in the surrounding locations. Diffusion will always continue until equilibrium is reached. Now, equilibrium, like the term sounds, is an equal distribution of molecules in different areas. So once equilibrium is reached, those molecules don't stop moving. They continue to move, but there will be no net movement because there is no longer an area of high or low concentration. Everything has equal or the same distribution or concentration of those molecules. So for example, if we have a red solute molecule, it is trying to evenly disperse itself or reach equilibrium in this solvent, which is represented by the blue molecule. So the solute is starting with an area of high concentration to the left-hand side of the screen. As time passes, it will continue to move towards the right, an area of lower concentration. Okay, in this far right picture, we have reached equilibrium. Those molecules are still moving, but there's equal concentration throughout. Now, there are 5 basic factors that are going to affect the rate of diffusion, temperature being the first one. So the higher the temperature, the faster something is going to diffuse. Now, this goes back to that particle theory of matter. The higher the temperature, the more energy all of those molecules have, therefore the faster they're moving, therefore the faster they can spread out or move away from each other to those areas of lower concentration. Surface area. Think about the example of trying to diffuse or dissolve or spread out a sugar cube versus granulated or a spoonful of sugar. The larger the surface area, the more surface area there is, the faster something is going to diffuse or to spread out. Because the more surface area there is, the more interactions there can be with the substance in which it is trying to diffuse through or spread out through. So the sugar cube has a relatively small surface area based on the volume, therefore it takes a long time to diffuse or to spread out in, let's say, a hot tea. Whereas a spoonful of granulated sugar has a large amount of surface area per volume, therefore it will diffuse faster or will spread out faster in that same hot tea. Concentration gradient. The higher the gradient, the faster something is going to diffuse. So that's saying that diffusion is always going to happen from an area of high to low concentration, but the bigger the difference between that high and low, the faster it's going to happen. So let's say if there's a 1% difference between high and low concentration, it's going to happen pretty slowly. But if there is, say, a 100% difference between high and low concentration, that diffusion is going to happen a lot more rapidly. Sizes of particles will also affect the rate of diffusion. The smaller the particles, the faster something is going to diffuse. Because the smaller particles move in and out of other molecules faster and they have the ability to spread out much or diffuse much faster than those large ones. They are less encumbered if they are smaller. The actual diffusion medium. Now, when I say medium, I'm talking about whether it is a solid, a liquid, or a gas. So a solid is going to have the slowest amount of diffusion because if we think about how those particles are packed in a solid, they're all very tightly packed and there's not a lot of wiggle room in between them or a lot of space for something to diffuse in between them. Whereas if you think about a liquid, those particles are moving past one another. There's a little bit more space and things can more readily diffuse or spread out within them. Gas, there is a lot of space in between all of those particles. Therefore, other molecules can easily spread in between them and therefore diffuse faster. So take a look at the following 6 diagrams, and I want you to pause the video and think about which molecules will diffuse in each of the figures and which direction will they diffuse in. Now, if we look at each figure has a membrane down the middle, note the size of the holes in between. Not all those molecules, red or blue, will always be able to fit through. So keep that in mind as you're trying to make your predictions. So pause the video now and determine which molecules will move which way and why. Let's see how you did. In the first situation, we're going to have the net movement from left to right. Those blue particles are going to be moving across to try to maintain or reach a state of equilibrium. The red particles, however, are too large to pass through, so they all stay on the left-hand side, even though they are not in a state of equilibrium. In number 2, similarly, the blue molecules or particles are going to move from an area of high concentration to low. Again, the red molecules cannot move from high to low concentration because they cannot fit through those spaces. In situation number 3, those spaces are slightly larger. The red particles can fit through, so therefore they are going to go from an area of high to low, reaching equilibrium, 3 particles on either side. In the case of situation number 4, those spaces are so small, those particles cannot move either way, regardless of the fact that there is a concentration gradient. Right? There's a higher concentration here than there is on the left-hand side. In the case of number 5, if we look at those particles are going to move from the right-hand side to the left-hand side. There were more particles on the left, or sorry, on the right rather than on the left. They're going to move from a high to a low area of concentration. In terms of number 6, the blue particles are already balanced. They're already in equilibrium. They're the same number on the left and the right. And whereas the red are too large to move through, so they cannot reach equilibrium. Here's a question for you. If you have raisins in your house, I want you to test this one out. Can you rehydrate a raisin? So based on what we just talked about in terms of diffusion and moving molecules, could you, in fact, rehydrate a raisin? Basically make it a grape again. Give it a try. See what you come up with. We'll discuss it in class. Osmosis is the next part of this lesson. It is a specific and special type of diffusion. It is when water diffuses across a semi-permeable membrane or a membrane that is selectively permeable. Now what that means is you have a barrier that will allow some things through but not others. So in the case of osmosis, water is allowed through but other larger molecules or charged molecules perhaps cannot get through. So let's look at this example down here on the left. Okay, we have a beaker, we have the selectively permeable membrane or barrier down the center. We have a high water concentration on the left-hand side and a low water concentration on the right. We have low sugar over here because there are no sugar molecules, whereas we have a high sugar concentration on the right because there are several sugar. Now, sugar cannot pass through this membrane. So think about, what do you think is going to happen? Well, what's going to happen is that water is going to move from its area of high concentration to the water's low area of concentration. Sugar is going to try to reach equilibrium, but it can't. So water tries to dilute and make sure that there's even concentration on both sides. Even though it's not possible, it is going to do the best it can to try to have equilibrium on both sides of that membrane. Okay, so in terms of biology, where we see this a lot is with the cell membrane or the plasma membrane that is surrounding all of the cells. Okay, water can pass through it. So it can diffuse through from areas of high water concentration to low water concentration, but large molecules like proteins and other molecules in your body cannot pass through it. So that is also why it is called selectively permeable. Okay? When we talk about osmosis, we have to keep in mind that there are 3 other terms. We have to think about what is happening with respect to the solution that a cell is in. So let's take a look at case number A. This is a hypotonic solution. Now, hypo means less than or a little bit. So hypotonic solution means that there is less solute or less dissolved substances in the solution than the cell that we are referring to. So when we use these terms, it's all referring to the solution in comparison to that specific cell. So if it is a hypotonic solution, there's less solute in it than in that cell. So we want to think in osmosis, it's going to move from an area of high water concentration to low. So if there is little solute, there is a lot of water out here. So high water concentration outside the cell. So it's going to move into the cell. There's going to be a net movement into that cell, causing that cell to swell. Now notice, water is going both in and out, but we want to know that net movement is into the cell. Okay, so that cell could actually, if it were in an actual human being, say a red blood cell, it could actually swell so much that it's going to burst, destroying the cell completely. If that same cell is put into a hypertonic solution, hyper meaning excess or a lot of, so that solution has more solute or more dissolved substances than the cell does. So what's going to happen is that water is going to try to balance everything out, all the concentrations. So that water is going to move out of the cell to try to kind of water down that solution. Now, as the water is moving out, that cell is going to shrink and shrivel up. Now, in biology, structure dictates function. So if its structure or shape changes significantly, it will no longer be able to function. So if this is a red blood cell, it won't be able to bind to oxygen any longer and carry that around to the cells, which is required for cellular respiration. Now, if we lastly take a look at this isotonic solution. So iso meaning same or similar. So this solution has the same or a similar concentration to the inside of that cell. So in this case, you have water moving both in and out, but there is no net loss or gain of water. Right? It is already in equilibrium. So let's take a quick comparison of our osmosis and diffusion. Things that are specific only to osmosis must be through a semipermeable membrane, and we are referring to the movement of water and only water. Okay, when we talk about diffusion, we're referring to all molecules, all particles, except for water. There's a specific word for that, and that's osmosis. Diffusion is looking at spreading things out over a large area, not just specifically through a membrane. Okay? The similarity between the two is there's trying to reach or create equilibrium, equal concentration on both sides.